Studies of the brain have focused on cells whose neural activity is easy to interpret. For example, place cells in the hippocampus are active when a mouse is in a particular location in an environment. Imagine you could hear brain activity, and each neuron corresponded to a note in a pentagram. The melody of walking along a path would sound like this. In more realistic scenarios, however, the hippocampus responds not only to position, but also to a variety of elements of the animal's experience, for instance, direction of motion or speed. In such a scenario, rather than beautifully arranged brain activity tuned to space, one typically finds a complicated mixture of activities which is harder to interpret in simple terms. The vast majority of the cells, the black nodes, encode multiple variables at the same time and enrich the representation of position, like harmony would give mood to a melody. To answer this question, we adopted recent experimental techniques that allowed us to record brain activity in freely moving animals with a head-mounted miniaturized microscope using calcium imaging. We injected mice with a virus carrying a fluorescent protein called GCamp and recorded the activity of hundreds of neurons in the Dentate, Gyrus, and CA1 regions of the hippocampus. Using machine learning, we trained a decoding model to read out the brain activity and obtain an estimate of the mouse's position at any point in time. We then tested the decoder in two ways. First, we identified place cells in the population and removed their activity from the data. Our model was still able to decode the animal's position without the place cells. To further understand how non-place cells contributed to position encoding, we shuffled the neural activity while preserving the overall structure, such that neurons could still play the same melody, but the harmony was rubbish. We then asked whether this had an impact on our model's ability to decode the animal's position. Shuffling neural activity had an impact on the decoder's performance. In other words, our decoder was indeed sensitive to the background harmony, as shuffling it increased the number of errors that it made. This suggests that looking at the brain as an ensemble of population activities may give us a much better understanding of how the brain represents and processes information.